Ever had that feeling in an exam where you know the answer, but you just don't get the full marks? Frustrating, right? Well, what if you could get inside the examiner's head and see exactly what they're looking for? Today, that's exactly what we're going to do. We're cracking the code on question 1 from an AQA GCSE Biology paper by using the official mark scheme. So, on the left, you've got the challenge, the June 2022 higher tier paper. But on the right, that's our secret weapon, the official AQA mark scheme. We're going to use this to literally pick apart question 1, showing you precisely how to tick every box and grab every single mark. All right, let's dive right in with the first part. This section is all about your core knowledge of reproduction. Let's see what they're asking. Okay, first up, question 01.1. You've probably seen a million of these, right? It's a classic complete the table task. You're comparing sexual and asexual reproduction, ticking boxes for 4 statements. It seems simple, but getting it perfect is all about the details. And voila! Here's the perfect answer, straight from the mark scheme itself. Let's walk through it. Row 1, cell division occurs. Well, that happens in both, so that's 2 ticks. Row 2, fertilization occurs. Nope, that's only for sexual reproduction. Row 3, genes are passed on. Yup, that's the whole point of reproduction, so that's a tick for both again. And finally, row 4, offspring are genetically identical. That's the signature of asexual reproduction. Get all 4 rows spot on, and that's 2 marks in the bag. Now listen up, because this is a golden nugget, a real piece of insider info from the mark scheme. It says, allow 1 mark for 1 or 2 correct rows. This is huge. It means even if you're not totally sure about the whole table, just getting 1 or 2 rows right will still earn you a mark. So the lesson here, always give it a shot. Okay, moving on to question 1.2. This one's a quick-fire recall question, nice and simple. Name the male gamete formed in flowering plants. And the answer is pollen or pollen grain. But here's where looking at the mark scheme really pays off. See how it also says to allow the answer nucleus in pollen grain? This shows you that sometimes there's more than one way to say the right thing. It's a great example of how the mark scheme gives you all the possible winning answers. All right, time to switch gears. We're moving on from pure memory recall to data interpretation. It's all about decoding this graph on meiosis to unlock those marks. So here it is, the graph for the next few questions. Before we even peek at what they're asking, let's just get our bearings. The y-axis on the left is the mean mass of DNA, and the x-axis along the bottom is time and hours. The line is just showing us how that DNA mass changes over time. Simple as that. So the first graph question, 01.3, asks, when is the DNA in the chromosomes being copied? In other words, where on this graph is DNA replication happening? So we need to be detectives here and look for the evidence. If DNA is being copied, its mass has to increase, right? And look, right there between 3 and 4 hours, the line shoots up, literally doubling from 4 to 8 units. That's our moment. That's DNA replication in action. And boom, there's our answer. The mark scheme confirms it between 3 and 4 hours. Just for spotting that trend, you get 1 mark. Easy peasy. All right, next puzzle. Question 1.4 gives us a helpful reminder. Cells divide twice during meiosis. Then it asks us to find the 2 times on the graph that show this division happening. So if we're looking for cell division, what does that look like on the graph? Well, if 1 cell splits into 2, the mean mass of DNA per cell has to drop by half. So we're hunting for those steep drops. And there they are. The first one at 5 hours, where the mass plummets from 8 down to 4, and the second one at 6 hours, dropping from 4 down to 2. Those are our 2 divisions. So the 2 correct answers straight from the mark scheme are 5 hours and 6 hours. You needed to get both of those right to get the mark for this question. We're on the home stretch now, just 2 more questions. These are all about applying what we know to the graph. First up, 1.5. What is the mean mass of DNA in arbitrary units in a sperm cell? Okay, so let's think. A sperm cell is a gamete, which is the final product of this whole meiosis process. If you look at where our graph ends up after that second division, it levels off right at 2 arbitrary units. So there's your answer. It's just 2. And for the final part, question 1.6. What is the mean mass of DNA in arbitrary units in each cell in an embryo? All right, let's put the pieces of the puzzle together.
An embryo is formed when a sperm cell and an egg cell fuse during fertilization. We just figured out that a gamete, like a sperm cell, has a DNA mass of 2. So when two gametes combine, 2 plus 2 equals 4. The DNA mass is restored to 4 arbitrary units, which, if you look, is exactly where her graph started. It all comes full circle.
And there you have it. That's question 1 completely deconstructed. So let's just take a step back and pull this all together. What are the big takeaways from an examiner's point of view for scoring full marks? Think of this as your personal checklist, straight from the examiner's playbook. To absolutely nail this question, you needed a few things. You had to recall core facts for that table, you had to carefully read the graph, you needed to spot the trends, the increase for replication, the decreases for division, you had to pinpoint specific points on the graph, and finally, you had to apply your knowledge of meiosis and fertilization. It just goes to show how one question can test a whole range of your biology skills.
What this really shows is that exams aren't just about memorizing stuff. They're about being able to apply your knowledge in really specific ways. So now that you know how to think like an examiner for this kind of question, what kind of question should we break down next?